is this virus that we're looking at?
Sure. So the virus that we're looking at in particular in this study is called 229E. It's sort of a bad name, but it's a alpha coronavirus, which means that it's sort of a distant cousin of the virus that causes COVID-19, although the symptoms that you get from 229E and other alpha coronaviruses that circulate in humans tend to be much more mild. It's similar to the common cold.
So yeah, is this the virus then that makes people sick in North Carolina with the common cold?
It's one of them, yeah. It's a little bit tricky because when we say common cold, generally people are referring to a set of symptoms as opposed to an individual virus. So they're talking about runny nose, cough, mild fever, and sort of general malaise. And those are symptoms that are characteristic of an upper respiratory infection that can be caused by any number of a wide variety of different viruses. But the viruses that my lab studies in particular are called alpha coronaviruses.
And so, from kind of reading over this and doing a little bit of research before hopping on here with me, it sounds like we've known about this 229 for decades, right? So, why has it taken so long to identify these antibodies?
That is a great question. And it was something when I started my lab here at Duke that it really sort of baffled me because it seems like now we have all these tools to be able to ask these questions, particularly in the wake of the COVID-19 pandemic, where we developed all of these new tools and technologies that now actually allow us to do this research. But the other really sort of more basic reason why we don't have this information yet is because oftentimes when researchers like me are trying to figure out how a certain virus interacts with the host cell immune system, or just the host immune system, I should say, is we'll collect samples from a patient who's either actively infected or who has recently recovered from a confirmed infection. And in the context of the common cold, that's a little bit tricky because most people will come down with these symptoms. They'll say, Shucks, I'm sick for the next week, but I'm not going to bother going to the doctor because this is probably just going to resolve by itself. They're just going to tell me to go home and get some rest. So it's actually sort of tricky to get our hands on these confirmed samples.
So, when you were doing this, what did you discover that is different than what we might have known previously or didn't know at all, I guess?
Yeah, it's sort of a case of didn't know it all. And it's interesting because there are, even though the disease is caused by the virus that causes COVID-19 and this 229E virus, even though the disease symptoms are extremely different, the viruses work in fairly similar ways. And so we're sort of excited to learn that antibodies that target a specific portion of this 229E spike protein, spike is the protein that covers the surface of the 229E virus and allows it to get into our host cells so that they can then replicate and infect. Antibodies that recognize a certain portion of that spike protein are capable of neutralizing the virus by preventing its association with our host cells. And we've seen very similar things in the context of COVID-19. So it's interesting that our immune system is capable of recognizing these two very different proteins and neutralizing these two very different viruses in similar ways.
And on that, I kind of want you to elaborate it. I was trying to come up with a way to visualize this to help the average person kind of understand this. Using like a lock and key analogy, how would you explain what this spike protein is versus what the neutralizing antibody is? How would you explain how they work?
That is very helpful. So to use sort of the lock and key analogy, I guess the spike protein would be the key that the virus uses to get into our cells. And there are receptors on our cells that would interact, that would act as sort of a lock. And these antibodies are blocking that key from getting into the lock, gumming up the works, so to speak.
So if the purpose, of these antibodies, or for, in our immune system, we create these antibodies and in theory they help us protect against future infections. So for someone listening to this who's like, okay, but if I'm getting sick all of the time, how am I still getting sick all the time if I'm still making these antibodies?
That's a great question. And that was one of the more interesting parts of the study for me, was because even though we make these effective immune responses, like you say, people continually get sick with these every few years. And part of the reason for that is because as our body learns to recognize these viruses, the viruses have to then evolve in order to keep infecting and replicating. That's how they reproduce. And so basically, you're trying to hit a moving target as this virus is constantly evolving. And eventually, the virus will evolve to the point where these antibodies no longer recognize that mutated spike protein, and you're susceptible to infection again. So in the future, if we were ever trying to develop targeted therapeutics or vaccines, we would probably need to sort of iteratively update those vaccines and therapeutics every few years so that we could recognize those strains that are now circulating throughout the population.
Yeah, looking kind of towards the future, how would this discovery, potentially impact future treatments or, to your point, eventual vaccines?
Yeah, um, these really potently neutralizing antibodies that we've isolated, we now know how they function. Again, it's by preventing that association between the lock and the key. And so our hope is that we can design vaccines that elicit antibodies that perform the same thing. And that way you'll have these antibodies circulating in your bloodstream. When you come into contact with these viruses, you'll be protected from disease and potentially even protected from infection. Um, like I said, that we're probably going to need to iteratively update these vaccines every few years. But the evidence is that these alpha coronaviruses mutate much more slowly than something like, say, the flu. So hopefully it won't have to be every single year.
And I'm glad you mentioned, COVID-19 earlier because a lot of people, I think, hear coronavirus and immediately associate it with that. And kind
Yeah.
of the vaccine discussion, obviously that was something that we saw a vaccine come out for in record time. So when they hear this, what's the realistic time frame? How far away are we really from a vaccine for the common cold, so to speak?
That's a good question. A lot of that Some of that is not necessarily a scientific question. Some of it comes down to how these vaccines get approved. We saw the approval of COVID vaccines in record time, in part because of the pandemic. There was such an urgent need for them. I suspect that we wouldn't see the same sort of rapid progression through the regulatory process for these common cold vaccines because, again, it's not addressing as severe a disease. But we have definitely learned a lot by going through the COVID-19 vaccine development process. And we are hoping we can apply those tools to these other more mild viruses.
And I'm reading the abstract, it highlighted that obviously the common cold, this virus, can be more dangerous for particular groups, our older population, younger children, etc. So how could this research potentially help protect those vulnerable communities?
That's right. That's a really important point because even though it's just a common cold for a lot of people, in infants, you have to worry about the development of croup, which can be more serious. And in the elderly, oftentimes these infections can lead to more severe symptoms like pneumonia. So we're really interested in potentially protecting these more vulnerable populations. It gets a little tricky when you're trying to develop treatments or vaccines for infants because obviously there's so much concern about making sure that everything is safe before you're administering those potential vaccines. And elderly, it's the restrictions are a little bit less difficult to overcome. I'm not sure how you would phrase that, but because you're not necessarily as concerned as you are with infants. That's
And
phrased very poorly, but I think you got my point.
yeah,
Sorry.
yeah. And it's kind of on that, a lot of us, right? We get a common cold, we have a runny, stuffy nose for a couple days, and then we kind of forget that we had it a few weeks later. But why is it still important from a research standpoint, to understand this virus and how it works, even if in the majority of people it isn't something super serious?
Yeah, so we learned a lot about the viruses like the virus that causes COVID-19. These are called beta coronaviruses. They tend to cause much more severe disease. But these alpha coronaviruses, which are of a different genus, we know very little about them, especially compared to the beta coronaviruses. And so there are alpha coronaviruses that are continually emerging into the human population. There are a lot of alpha coronaviruses in animals that we're worried can potentially cross over into humans. And those might someday cause more severe disease, something more severe than the common cold. So the hope is that by studying these viruses that cause more mild disease, where we have a lot of potential patient samples, a lot of resources that we can use to study these sort of fundamental processes, we can learn more in case there are future pandemics.
Is there anything that the public can do to help you in your research right now?
That's a good question.
I
I'm,
didn't know
I'm
if you were actively looking for, samples or anything like that, or
so I would like to be able to say that, but I'm not a medical doctor, and so I'm not the one who's actually collecting the samples. So I would probably say no, other than Wash your hands and cover your mouth when you cough.
It's sage advice. I guess just to, before I let you go, I want to ask, obviously the point of research, right, is to go in with a question and see whether or not your hypothesis is correct or not. So what was that question that you went in with? And you've kind of mentioned a few things throughout this of things that were maybe surprising to you, but is there one thing that stands out to you is the most significant?
Most significant. That's a really good question. I think maybe the most significant finding, which again, I was not necessarily expecting going into this, was the finding that these antibodies sort of lose recognition of previous strains. To phrase that another way, we were able to test different historical strains of this 229E virus, spanning back all the way to the 1960s. And we found that these antibodies, even though they really potently neutralize contemporary strains, so things that might be circulating today, about 8 years prior, you start to lose that efficacy. So the virus is gradually evolving over time, and that does have an impact on the immune responses that we make.
Awesome. Is there anything else that I didn't ask you, Daniel, that you want to mention that's pertinent to this or your hopes as to, I guess, where you go from here?
Well, I mentioned that um, the hope is that we can take this information and use it to design therapeutics. That's going to be a big focus of my lab moving forward. But another thing that I want to mention, a lot of this work was done at Duke, but it was also the result of a really fruitful collaboration between Duke and some world-class virologists at UNC. So it's really cool that we can work together and have all this groundbreaking research happening right here in the Triangle.